Hello, and welcome to Molecular Biology of the Coronavirus. My name is Nathan Peabody, and this lecture is intended for bioscience at Merritt College. We've created this online set of modules to replace our in-class lectures, and I hope this lecture gives you some valuable information and helps you understand just what is exactly happening in the virus and how it takes over the host to create more virus particles. Let's start off with what exactly is a virus. This slide and many slides during this lecture will be from Professor Raccaniello at the University of Columbia, who has very kindly put all of his lectures this semester of virology for free on YouTube. So I encourage you to go to YouTube and check out his lectures if you want to know more about viruses. Viruses vary a lot, and they can have a variety of different components. In general, they're all parasites that infect their host in some sort of obligate intracellular way. They can have different types of genetic material, whether it's DNA or RNA, and that can either be plus or minus, and it is often surrounded by a protein coat and also sometimes a membrane. Here in this diagram, you can see many different types of viruses, and let's go through some of them. Down here at the bottom is our coronavirus, the one that we're going to currently talk about in this lecture, and it has a membrane with a bunch of proteins studded in it and RNA inside. Above left of the coronavirus is a polyhedron type of virus. This virus does not have a plasma membrane or cell membrane. It's simply a bunch of proteins forming a polyhedron type of structure with also a bunch of spike proteins sticking out of it. And by comparison, up here at the top, we have a virus that has a polyhedron surrounding a DNA in the middle, and then a protein-studded plasma membrane up at around the whole thing. And this, of course, is a cutaway. Some of the other polyhedrons are solid without a cutaway view, and you can't see their DNA. During the course of this brief lecture, I'm going to show you many different types of images. Images such as the ones that you see here are all drawings, most likely made by a researcher or a PhD or maybe an MD or someone else in the laboratory that has been trying to draw something very accurate using all the data available. So when you see a cartoon like this, you should assume, especially because it comes from Dr. Raccaniello's lab, that it is all entirely accurate and scientifically valid, and that the different structures you see are all representing something very specific to the best of the ability of the author. Later, I'm going to show you actual pictures from various types of microscopes and also other drawings from other artists, and we're going to go through them. Here's a picture submitted by the National Institute of Allergy and Infectious Diseases for the current coronavirus. It was taken with a transmission electron microscope, and they estimated that the size, the diameter of the virus is about 70 to 90 nanometers. Other laboratories are thinking it might be a little bit bigger. One thing to note is that these structures look very blob-like. Some of the other viruses in the previous picture, especially the polyhedrons, may be very specific in their structure, but this, because it's a membrane, you might think it's a little more blobby and mobile. So even though they generally are a sphere, these pictures like this show that the actual structure of the virus might vary just a little bit. Here we compare the size of the coronavirus to other various proteins and cells. At the top, you can see the different types of machines capable of taking pictures, and light microscopy can't quite see the coronavirus. However, there are viruses that are big enough to be seen under normal light microscopes. So for the coronavirus, we need to have an electron microscope. Here's a nice 3D image created by Fusion Medical Animation, and you can see the nice protein studded on the surface. Here we have an image released by the CDC and their illustrators, Alyssa Eckert and Dan Higgins, of the SARS-CoV-2 virus. And you can see in gray is the plasma membrane, the lipid bilayer, and inside of that would be the DNA, which we don't see here. And then on the surface of the virus would be the various proteins. There's the triangular red protein, that is the spike protein, and then there are a couple other different proteins embedded in the membrane. Let's take a moment to appreciate this picture, the illustration that they've created. And For example, parts of it are out of focus, which of course they're creating it on Photoshop, so they don't need to have anything out of focus, but they do that for a nice effect. And then also you'll notice the scattering of other orange reddish particles around the picture. But these other viruses off in the distance that seem too small imply a sort of, give an emotional response. You're surrounded by these little tiny things that you can't see and they're off in the distance. And then one huge one up close. All effects that they did to convey something about the virus to humans, while also showing important structures. Here we see the plasma membrane in blue, and the spike protein is embedded in that plasma membrane. In addition to the spike protein, there are three other proteins in the plasma membrane: the envelope protein, an HE protein, and a membrane protein. All right, let's talk about exactly what does that membrane protein do. So let's rewind a little bit. Here is the RNA, the plus-stranded RNA on the inside of the SARS-CoV-2 virus. That RNA is encapsulated by another protein. The RNA is not just loose on its own. It is believed to be single-stranded plus RNA, but it has a whole bunch of proteins that it is looped around and encapsulate it and package it. And that packaging protein is called a nucleoprotein. And when you put the nucleoprotein and the RNA together, that becomes a nucleocapsid. So in the middle of the virus is a nucleocapsid, and it's surrounded by a plasma membrane. And the plasma membrane has 4 different proteins in it. The other 3 we already talked about. This 4th one is the membrane protein, being drawn here in green. And the membrane protein is believed to associate with the RNA on the inside and either holds it in place or keeps it organized or protects it. Whatever the exact function is, this membrane protein connects to the nucleocapsid, keeping the RNA and the nucleoprotein inside of the membrane. Here's a nice picture showing all the proteins we just talked about. You have an enveloped RNA virus that has a plus strand of single-stranded RNA inside. This RNA is looped around nucleoproteins or bound to it, and that becomes the nucleocapsid. It's surrounded by a plasma membrane, a lipid bilayer, and the lipid bilayer has 4 different proteins in it: a hemagglutinin, a spike protein, a membrane protein, and an envelope glycoprotein. Okay, let's focus in on the spike protein and just what it does. It's an area of active research because this is the protein for which we will make antibodies to in our own body if we successfully fight off the disease. And it's also the area where we are going to be looking at for different types of drug therapies to maybe prevent this thing from gaining entry into cells. Here's a close-up of the spike protein, and you notice it looks like a triangle, and that's because it has 3 identical subunits, which are all bound together to form one functional spike protein, which means it's a trimer. 3 identical units forming one. When the protein, the trimer, is first made, it's inactive, and it requires cleavage by another protein called furin. Furin is made in the host body, in our lung cells, and it has some other physiological process for which we use it for. So here's a cartoon of the spike protein. You can see the general cell cycle on the bottom left, which we'll talk about in a bit, and then above it is the virus. And then in the middle and on the right side is the actual protein. And the protein is all colorful because these are different ways that the protein folds upon itself. And the schematic above shows the different regions on this protein. The protein you're looking at there is the previous SARS-like COVID virus that was in 2013. And this virus was not nearly as lethal as the SARS-CoV-2 that we are currently experiencing an outbreak on. And the reason is because the arrows are pointing to this S1, S2 site. And in the current COVID-2 variety, there's a furin cleavage site there. And this furin cleavage site allows the spike to be activated by a wide variety of cells. And furin is very common in lung cells. The spike protein is a hot topic because it's believed to be 10 to 20 times more efficient at gaining entry into human cells than the SARS virus from 2002. So it is the source of our antibodies and what future vaccines might be made of, antibodies to this. It may be the reason that the virus is so contagious and spreads so easily. Okay, let's talk about how the virus uses the spike protein to gain entry into the lung cell. On one hand, the spike protein is going to connect to this ACE2 receptor, but there's also another protein in the lung cell called a TMPRSS2. And these are two proteins that we make on the surface of our lung cells that are being used by the spike proteins on the virus. And there may be more cells, more transmembrane proteins on the lung cell that are also being used. But the ACE2 receptor and the TMPRSS2 are cellular components that we already have. And what happens is that the spike protein will connect to both the ACE2 and then another spike protein will connect to TMPRSS2. A hot topic in research right now is the furin molecule. It's a transmembrane protein. It's not shown here in the drawing, but it should have a section going through the host lung cell, anchoring it into the membrane there, the lipid bilayer, so it won't float around. But when the spike protein from the virus gets near the furin, there are cut sites on it, get cut or cleaved by furin, and there should be 3 different cleavage sites on the spike protein, presumably, for the trimer. All of the proteins shown on this diagram are all anchored in the plasma membrane. They're all transmembrane proteins. So really the virus and lung cell are very close together. Everybody's close enough to be activated and then latch on or bind to somebody else. The activated trimer spike protein will then be able to bind to the ACE2 receptor and change its conformation. And now we get a chain reaction as energetically favorable shapes start appearing all over the lung cell and the virus. So the ACE2 receptor recruits the TMPRSS2 transmembrane protein, and then more and more spike proteins start reacting with more and more of these 2 receptors, and then sooner or later you have endocytosis, and the whole virus is pulled into the cell by all of these transmembrane proteins acting in some sort of mechanical cohesion to bring in the virus. Let's move on to exactly how does the virus get into the cell and what does it do once it's inside the cell. Here's a list of all of the single-stranded RNA plus sense viruses. Well, not all of them, but some. You can see in the middle is our coronavirus, and there's other viruses that you may have heard of. Now, of course, there's also DNA viruses and negative sense RNA viruses, but coronavirus is a single-stranded plus sense RNA virus. Let's start with a stereotypical plus sense single-stranded RNA virus, the poliovirus. Now, when it's plus sense, it means it's actually an mRNA. And usually they also have a cap on it and maybe a poly-A tail for those of you that know genetics. But in general, what happens is the poliovirus gains entry into the cell. It releases the RNA, which is basically mRNA. That mRNA then makes its way into the machinery of the host cell, translates the mRNA into protein, and then the protein is cut up and made into a bunch of subproteins. At the same time, the mRNA needs to duplicate itself. The mRNA duplication steps are shown in numbers 8 through 11. And because the mRNA is not DNA, nothing needs to happen inside the nucleus. This is all outside the nucleus in the cytoplasm of the cell. And the mRNA is plus strand, so in step 9 to 10, it needs to make a negative sense copy of the mRNA so that it can make more plus strand copies of the RNA. And then it continues this duplication effort, and eventually then they have enough RNA that it can package it into new virus particles. The new virus particles are packaged in steps 12 and 13, where a bunch of proteins made from the mRNA, VP0, VP2, and VP1, are used to create the polyhedron around the mRNA in the middle, which also probably has some nucleocapsid packaging. And then the virus is ready to be spit out by the cell. I wanted to show you the poliovirus first because that seems like a very logical way and simple way, if you have plus-stranded mRNA, to make a bunch of proteins, duplicate your mRNA, and then package everything back into a bunch of new viruses, which are then exocytosed by the cell. Coronavirus, for whatever reason, is different. And let's go through this diagram. We'll go through it step by step. Let's start up at the red arrow, number 1. The coronavirus comes in, as we talked about, and has its spike protein, the trimer, gets activated by furin, and that furin attaches to several membrane brown proteins on the plasma membrane of the cell, and then it is endocytosed. It's brought inside. And you'll notice there's a cell membrane layer from the cell around the entire virus. This is then moved into the number 2 spot. At number 2, you can see the mRNA is translated by the host cell ribosomes and made into a few proteins. At number 3. Here's where things get a little wacky. It seems inefficient, but this is the way the coronavirus works. It makes these double membrane vesicles, and then inside these vesicles, it starts producing a bunch of different sized mRNAs. And these different sized mRNAs then can produce a variety of different proteins. And also and that is happening in 4 and 5. Step 7 in the double membrane bound vesicles, it also creates a bunch of entire RNA genomes to be packaged later. Here at step 5, we can see a big difference between the coronavirus and the poliovirus. The poliovirus just made one long translation from its mRNA. It had one long protein, a proprotein, and that could then be cut up into a bunch of different subproteins. Here, the coronavirus has a bunch of different sized mRNAs, and those mRNAs code for the various proteins that you see down below it. Now, as I mentioned, the genome, the plus-stranded RNA of the coronavirus, is also being replicated. So somehow, obviously, it needs to keep it all straight because it wants to only package the entire genome of the long strand of the mRNA. It doesn't want to package all the little mRNAs shown in parts 4 and 5. So in part 7, it's replicating the entire genome. It has to also put a cap on it. And then this genome goes down into 8. In 8, a bunch of the nucleoproteins that have been translated from the mRNA combine and wrap around or connect to the RNA in some way to form the nucleocapsid. And this nucleocapsid is then ready to be surrounded by a membrane studded with the 4 other proteins we've talked about earlier. Now, the 4 transmembrane proteins that are in the plasma membrane or lipid bilayer surrounding the coronavirus are all being translated and studded into membranes at the red arrow, part 6. This, of course, is at the ER, the endoplasmic reticulum, outside of the nucleus. And this is where the virus gets its plasma membrane. The plasma membrane comes from the host, and the virus cannot make it itself. And here is the final step where the nucleocapsid that has both the nucleoprotein and mRNA is then surrounded and enveloped by a plasma membrane studded with the 4 various transmembrane proteins. And this whole virus particle seems to go inside of a vesicle, so it's surrounded by another plasma membrane, and this plasma membrane vesicle then gets exocytosed from the cell. I'd like to point out that this exocytosis of the virus outside of the cell, and also the endocytosis for the virus gaining entry into the cell, is all probably mediated by different cellular components that the cell already has that the virus has hijacked. And viruses do this. They hijack cellular processes, use them to replicate their genome, make their proteins, and then assemble new viral particles, all just taking over the host machinery. Let's take a look at the virus particle and the RNA that comes with it and all the different proteins that are made from it. Now, we've talked about 5. 4 transmembrane proteins that stud the outer plasma membrane of the virus particle, and then also the nucleoprotein, which wraps the RNA and packages it and coils it. But there's also a whole lot of other proteins made from the mRNA of the coronavirus. Let's dive into that for a little bit. Early on, when the Chinese realized that they had a novel virus going around, one of the first things they wanted to do is sequence the RNA of the virus and publish it so they could aid researchers in developing therapies and resistance to this coronavirus. And what they found was that the RNA of the coronavirus is around 30,000 base pairs long. They say that 30 kb. Let's analyze that 30 kb genome here in a picture from the New York Times, which I encourage you to go read on the internet. The reference link is supplied in the video. Open reading frame 1AB is the very first thing that's translated when the virus enters the cell. The mRNA immediately produces this really long protein, 1AB, and then also a shorter one, 1A. And this big protein, or the two proteins, are then chopped up into a whole bunch of little proteins that start doing a whole bunch of other functions to mess up the cell. Then later, other mRNAs are made for the structural proteins you see on the right. All right, let's talk about the SARS-CoV-2 genome from the diagram you just saw. So it's 30,000 base pairs, which means it's 30 kb in length. It's single-stranded RNA. And open reading frame number 1 is one long protein, which means it can be cut into smaller functional proteins. And 16 proteins are cut from this one open reading frame number 1. The first one I want to talk about is NSP1. NSP1 is about 180 amino acids long and is a sabotage protein. It hinders the host cell's ability to make its own proteins. Another interesting protein is NSP4. NSP4 is about 500 amino acids in length, and it is called a bubble maker protein. It helps build the strange double membrane vesicles where viral replication of the RNA will take place. Another interesting protein is NSP10. It is about 139 amino acids in length, and it is a genetic camouflage protein. It serves to hide the virus genes from the host. Host cells often have a lot of antivirus protection, sometimes proteins that look for virus RNA and cut them up. And NSP10 somehow serves to hide the viral DNA from the host protection proteins. And before I forget, let's do a quick review of genetics. If there are 30,000 base pairs of RNA in the COVID-2 genome, that translates to 10,000 amino acids. You need 3 base pairs of RNA to code for 1 amino acid. Now let's talk about 2 of the proteins we talked about earlier, the spike protein and the nucleoprotein. The spike protein, which may be translated slightly different than open reading frame 1, it is coded for with 3,830 RNA base pairs, which means the protein is about 1,275 amino acids in length. And this is a trimer. So the host cell will make 3 of these individual spike proteins that then bond together and they form a functional spike protein. And then this spike protein, which is in the membrane, needs furin to come over and activate it. And the furin will come from the host cell. And the last protein I want to talk about is the nucleoprotein. The nucleoprotein is 1,260 base pairs in length for the coding region, and that translates to about 420 amino acids. And the nucleoprotein associates with, bonds with the RNA in the COVID-2 genome, and the nucleoprotein plus the RNA, when formed together in their helical manner, becomes the nucleocapsid. That concludes module for the SARS-CoV-2 molecular biology, and stay tuned for references that might be useful on the quiz.